So under chapter 6, we have 3 subtopics. So 6.1 is all about introduction. We are going to be looking at mutation, classifications and types. And then 6.2 is about gene mutation, and 6.3 is about chromosomal mutation. So mutation refers to permanent change in the nucleotide sequence of an organism's DNA. DNA bukannya RNA, and therefore dia akan create genetic diversity. So kenapa dia boleh create genetic diversity? Because now we have new DNA composition. Sudah berbeza dia punya kandungan DNA. So mutation adalah yang kita panggil sebagai event dia. Dia adalah sejenis phenomenon. It is something that happened. So when mutation happen to an organism, organism itu ataupun gene itu kita panggil dia mutant. So mutant is a mutated gene or an organism carrying a gene that has undergone a mutation. So two almost similar terms but very different. So the first type of mutation is what we call spontaneous. Spontaneous meaning it's random, maksudnya dia unprovoked. Maksudnya it happens in nature. We didn't do anything, it happens on its own. So normally it happens during DNA replication. Okay, DNA replication, this is kita punya S phase. Also during gamete formation. So gamete formation refers kepada meiosis. When it happens in gamete formation, normally we call it non-disjunction. Non-disjunction refers to the failure of homologous chromosomes or sister chromatids to separate properly. Okay, so this could be either meiosis 1 or meiosis 2. So as a result, it will produce abnormal number of chromosome in a gamete that is passed down to zygote. The second type of mutation is induced mutation. So this is contrary to the first type. With induced mutation, we induce the mutation using mutagen. Kita suruh mutation itu berlaku. So mutagen kita ada dua jenis. Kita ada mutagen yang physical, kita ada yang jenis chemical. So kalau physical ini seperti UV rays ataupun gamma rays ataupun X-rays. Kalau chemical agent pula, kita boleh ada colchicine ataupun ethidium bromide. Okay, contoh pertama yang kita ada di sini adalah colchicine. So apa yang colchicine buat adalah dia menyebabkan mitotic spindle gagal terbentuk. So yang ini merujuk kepada kita punya spindle fiber. Spindle fiber ini, as we know, fungsi dia adalah to separate sister chromatid ataupun homologous chromosome. So kalau kita gagalkan pembentukan spindle fiber, maka kita tidak boleh separate mereka berdua ini. Okay, so kalau kita lihat di dalam rajah ini, sepatutnya seperti inilah proper disjunction. Maksudnya satu sister pergi ke sini, ke kiri, satu lagi sister pergi ke kanan sini. However, disebabkan colchicine ditambahkan kepada sel ini, maka spindle fiber tidak terbentuk dengan elok. Maka kedua-dua sister chromatid ditarik ke kiri. Contoh yang kedua, kita ada ethidium bromide. So ETBR. So ethidium bromide ini adalah kristal yang mana akan masuk di celah-celah DNA. So dia akan menyebabkan stretching of DNA, dan bila berlaku stretching, kita akan prone kepada insertion of extra DNA base during DNA replication. Maka kita akan bertambah DNA bases. Okay, gene mutation juga dikenali sebagai point mutation. So gene mutation refers to a change in a single nucleotide pair of DNA in a gene sequence, meaning it refers to A that will be paired with T, or it refers to C that is paired with a G, or vice versa. So, dia hanya merujuk kepada satu sahaja. Di dalam kromosom kita yang banyak-banyak ini, it refers only to this one tiny bit. Okay, however, dia punya akibat adalah menyebabkan perubahan daripada bentuk sekuens mRNA yang dihasilkan. So, bila kita ada DNA, kemudian kita buat transcription, maka kita hasilkan short mRNA strand. So, kalau mRNA ini sudah transcribe kawasan DNA yang kena mutation di sini tadi, maka mRNA-nya juga akan berubah sekuens. So, akibatnya dia akan menyebabkan perubahan dari segi amino asid yang kita akan hasilkan. So, kalau kita ada perubahan susunan amino asid, maka eventually kita juga akan mendapat different types of protein. And if this kind of mutation occur in a gamete or cell that will become gamete, obviously this will be inherited down the generation. So we have 4 major types of this kind of mutation. So we have substitution. So substitution maksudnya kita tukar, gantikan. And then we have inversion. Inversion maksudnya kita songsangkan, terbalikkan. And then we have deletion. And we have insertion. Look at an example of a DNA. So this is what a normal DNA will look like. So we have 5' AGC TTA 3'. So obviously because this is DNA, it must be double-stranded DNA. And it must also have anti-parallel arrangement. So the first example is base substitution. So base substitution means we exchange. So in this pot we are supposed to have a T. So we were supposed to have a T and the base pair is supposed to be an A. So instead we have G and C. Inversion obviously requires more than one pair because we need somebody for us to exchange it with. So in this case, we were supposed to have CT, but we already exchanged it. So now we have AGTC, TA. So obviously, dia punya strand yang anti-parallel juga berubah. Sepatutnya kita ada TCG AAT. Sekarang kita sudah ada TCA GAT. Okay, yang seterusnya kita ada base deletion. So base deletion, as the name suggests, meaning we just remove a pair of DNA base. So we were supposed to have AGCTTA. So we already removed the T. And obviously on the other strand, on the opposite strand, the A is also removed. Okay, in contrary to base deletion, we have insertion. Insertion meaning we add a new pair or more than one pair. So now we have 7 pairs of DNA bases. So all 4 types of gene mutation can lead to these 4 consequential mutation. So ini yang saya panggil consequential mutation. Kenapa? Sebab ini adalah akibat. Ini adalah result daripada keempat-empat mutation yang awal tadi. So ini adalah pattern yang biasanya kita akan observe. Kalau kita ada base substitution dan inversion, selalunya kita akan mengakibatkan berlakunya missense, nonsense dan juga silent. Tetapi kalau mutation yang berlaku adalah insertion atau deletion, it will always lead to frame shift mutation. In missense mutation, the mutation will cause changes in one amino acid within a protein. So mari kita lihat example. So this is the original gene. So no mutation happens in this. So now we have G. Base substitution because we change A for a G. So now instead of getting TYR amino acid, now we get CYS. Okay, so one amino acid is changed. In nonsense mutation, nonsense maksudnya merapu. So kenapa dia merapu? Because perubahan codon tadi menyebabkan munculnya stop codon. So when stop codon suddenly appear, it will cause a premature translation stop signal. So look at this example. So we were supposed to have UAU here, but the U has been substituted into a G. So now we have a UAG. So UAG is one of the 3 stop codons. So kita ada lagi yang UAA, kita ada lagi UGA. So now it will cause the translation to stop. So akibatnya yang lagi 2 ni, kat sini sepatutnya ada lagi 2 amino acid, sudah pun hilang. And finally, we have a silent mutation. So silent mutation, it does not cause amino acid change within a protein. So kenapa boleh berlaku seperti ini? Because one amino acid boleh dihasilkan oleh beberapa codon, bukan satu sahaja. So contohnya seperti ini, kita ada mutation di codon ketiga, sepatutnya kita ada UAU, tetapi kita sudah substitute menjadi UAC, tetapi amino asidnya masih yang sama jenis. So sebab itu kita panggil dia silent, sebab macam tiada apa-apa yang berlaku. Okay, one of the most popular example for base substitutions in gene mutation is this disease called sickle cell anemia. So sickle cell anemia, we know, is when a normal red blood cell, which is supposed to be biconcave, is failed to be produced in that shape. So instead of that, we have a resulting sickle cell shape. And because of that, we have lower oxygen transport. So because of that, it will lead to anemia. Okay, so this is caused by glutamic acid. Glutamic acid, the codon is supposed to be GAG or GAA, so 2 options. However, these codons have been changed, have been substituted into valine. So valine is either GUG or GUA. So this mutation occurs in one of the beta strands of the hemoglobin of the red blood cell. So the effect of a person with sickle cell anemia includes anemic or stiff hemoglobin, or we could also say sticky. So therefore it tends to accumulate in capillary, causing blockage. And then obviously hemoglobin is not efficient in transporting oxygen, and therefore it will cause fatigue. In frame shift mutation, however, the mutation is slightly more virulent. Dia lebih aggressive. So this mutation involves insertion or deletion of one or more base pairs in the nucleotide sequence of DNA. So let's have a look at the example here. This is the normal code. So event pertama adalah if we delete G. So if we delete G, so we were supposed to have GAG GUU, but now the G is deleted. So now we already move our codon reading frame. Okay, codon reading frame. So we know we read codon in triplets begini. So now that 1 is already gone here, G itu sudah hilang, maka kita akan ubah kita punya reading frame jadi begini. Okay. So bila kita ubah begini, maka pembacaan codon akan berubah bermula dengan UUC, CUA, AAC, CU, yang ini tambahan di sini A. Okay, akibatnya all of the amino acids will change. This is what we were supposed to get, but this is the resulting. So all of the amino acid semuanya sudah berbeza-beza. Okay, so kalau kita ada insertion, so insertion of G, so sepatutnya kita ada 1, 2, 3, 1, 2, 3, 1, 2, 3. So sepatutnya kodon keempat ini bermula dengan AAA, tetapi sekarang ada G yang baru wujud di sini, maka kita ada GAA, ACC. So, sekarang juga berubah amino acid yang terhasil. So, basically di mana berlakunya mutation, di sebelah belakang dia onward sampai hujung, semua amino acid dia akan berubah jenis. Kenapa? Sebab dia punya codon reading frame sudah berubah.